Hello everyone, this is a tutorial for Aneuvis, which is a web tool for analyzing chromosomal number variation in single cells. The goal of Aneuvis is to take in copy number data obtained from either FISH, single-cell whole genome sequencing, or SCI experiments, and to output a summary of the numerical chromosomal changes that might have occurred between experimental groups. Here I'm showing an overview of how Aneuvis works. Essentially it works downstream of many existing frameworks which are used to quantify the number of copies of each chromosome. So here we have copy number matrices and we have knowledge of experimental groups, and Aneuvis integrates the knowledge of experimental groups with copy number matrices to generate a series of summary statistics, which can be found in the table output, as well as the set of visualizations, which are all calculated automatically. So we have some documentation. We have frequently asked questions, as well as a email that you can reach out to with any suggestions. So the first step in Aneuvis is to upload data. Here I'm going to upload FISH data from 3 different treatments. I'm also going to upload some single-cell whole genome sequencing data that has been processed previously using Ginkgo. And so the first summary that we have here is the table summary. We have within this table a set of statistics that quantify the degree of numerical variability within our populations of cells, and we see that they're divided by both the treatment type as well as the method used to quantify the number of chromosomes. This table is also sortable and filterable. So if we like, we can filter, for example, only those numbers that occur within a particular range. We can also download the statistics table if we like, and we have some additional information on the how these statistics in particular were calculated down below. We have a similar table grouped by this time by chromosome as well, and we have the same interpretation down here. We also have a chromosome level summary that can be downloaded, as well as a platform concordance test, which essentially is looking to see between any 2 experimental platforms that are uploaded, how often do the values that are observed change together. The details can be found here if you're interested. The next tab is the visualization tab. This is the second out of the 3 main tabs. We have some scatter plots in this case of some of the scores that were generated from the table summary. These scatter plots are also interactive, so if you're curious about which value in particular this circle corresponded to, you could highlight it and you could get some more statistics from that particular sample shown here below. We also have a ternary plot showing the proportion of diploid, polyploid, and aneuploid cells by group. And similarly to how we did in the table summary, we also have the scatter plot grouped by chromosome, and it's also interactive. Now we have some data type specific visualizations. We can see here are some grid plots which are specific for FISH data. We also have single-cell whole genome sequencing heat maps. If we had SCI data, we'd have a heat map for that as well. The last tab corresponds to hypothesis testing, and we can perform tests for individual data types. For example, just for the fish data, we can ask whether our experimental groups differ in the degree of numerical chromosomal variation or aneuploidy. So what we'd like to do is, in this case, select first the number of permutations, then we select the score that we'd like to permute. In this case, we're permuting the aneuploidy score, and then we can hit the permute button, which will generate a permutation for us, and we'll ask the question, or answer the question, is there a difference between any of the pairwise treatments shown here? And there's an interpretation of the table columns that is shown down below, as well as some details on the methods of how to interpret this plot. And we have that for every available data type, as well as a multi-platform summary, where we can get an overall summary integrating all of the data types that were uploaded. Given that the treatment groups were the same, is there an overall difference in terms of the degree of aneuploidy that we see? So thanks for watching, and good luck with your analysis.